Hello everyone and welcome to our webinar today. My name is Mark Lynch and I direct the Americas Microfluidics Sales and Support Organization. Today I'm here to talk to you about genomics workflow solutions leveraging microfluidics. Today's session is going to last around 40 minutes with 5 minutes for questions and answers at the end. We appreciate your attendance and hope you find the session interesting. The agenda for today's session is to start with an introduction to the Fluidigm microfluidic technology. And what that means is the Fluidigm integrated fluidic circuits, or IFCs. We're going to talk about how these IFCs work and what types of applications they can be used for. In our introduction, we'll also cover off on the systems that are used for genomic applications. Our IFCs run on instrumentation, and we'll introduce what that instrumentation is and how it fits in the genomics workflow. The second part of today's presentation is to introduce our new Advanta RNA-Seq NGS library prep kit. This is a prep kit that can be used to prepare libraries for next generation sequencing, in particular RNA sequencing. We're going to start with the system overview, the workflow overview, and then show you some of the data that one of our customers at the University of North Carolina has recently generated using that new system. So again, we hope you find today's session interesting and informative. So let's start by introducing the Fluidigm microfluidic technology. What does this actually mean? So if you take a look at the next slide, what you'll be able to see in a few moments are all of the different parts that come to make the puzzle of microfluidics. Microfluidics itself is really focused on genomics-based applications. And so let's start with the systems shown on the top left-hand side of the slide. The instrument systems that we will talk about today include the Juno system and the Biomark HD system. The Juno system is our preparatory device. This device can process any of the Fluidigm IFCs. That's important because it means that all of the workflow is pretty much automated using the Juno system and using our microfluidic devices. The other platform that we're going to introduce to you today is the Biomark HD platform. This is shown the tall blue instrument behind the Juno and the C1. The Biomark HD system is our microfluidic real-time PCR system. Whatever applications you do using real-time PCR can be applied to the Biomark HD platform. But all of the applications will be running in microfluidic reaction volumes. And this is key. It's key because these microfluidic reaction volumes help you to improve reaction efficiency while reducing the cost per data point. So these systems all work with our integrated fluidic circuits. And on the right-hand side of the slide, you can see one of our integrated fluidic circuits. This IFC, or integrated fluidic circuit, is about the same size as a 96 well or a 384 well plate. It means that if your lab is used to running 96 or 384 well plates, that these IFCs can be used with your existing workflows. There's no need for any specialized equipment to process these IFCs. What's more is that the microfluidic nature of our integrated fluidic circuit means that you can process tens to thousands of samples at nanoscale reaction volumes inside this integrated fluidic circuit. And the integrated fluidic circuit shown on the right-hand side of the slide runs on the instruments shown on the left-hand side of the slide. In addition to the hardware and the consumable, the hardware being the instrument and the consumable being the integrated fluidic circuit, we also offer a range of different reagents. This is because, as you'll see in a few slides' time, we've got various different applications that we use. And so Fluidigm also provides, as part of the workflow, a lot of different reagents, not just for controlling the integrated fluidic circuit, but for running the enzymatic parts of the reactions as well. Examples of this include the Fluidigm reverse transcription reagents and the Fluidigm preamplification reagents. Lastly, because our system is flexible, we have an assay design service also. The purpose of the assay design service is so that you can easily design, define, and develop assays using microfluidics. You'll see later on in the presentation that a lot of our microfluidics, or in fact all of them, are not pre-spotted with content. That gives you total flexibility to the workflow. And so the D3 design process enables you to take markers that you're interested in studying and customize them on the fly. That's not possible with any other type of solution. And so the Fluidigm Genomics products, really related to microfluidics, are system solutions driven by automation and microfluidics. So let's take a look at the IFC in a little bit more detail. The IFC shown on the right-hand side of this slide is an example of our 48.48 IFC. The LP at the start stands for library preparation. In this IFC, you can see that we have 48 sample inlets and 48 assay inlets. And the way in which we parallel load those IFCs mean that every single sample on the left-hand side sees all of the assays on the right-hand side. So you end up with a matrix of 48 by 48. And that means on one of our LP 48.48 IFCs, you're able to generate more than 2,300 individual PCR reactions. That is a scale that's very difficult to achieve on standard 96 or 384-well plates. Further to this, because of the nanoliter reaction volumes that take place inside our IFCs, you're able to achieve greater cost effectiveness. This is because you're taking volumes that are traditionally very large and putting them down into nanoliter reaction volumes. In addition to this, the labor is much more cost effective using microfluidics than using 96 or 384-well plates because the workflow is pretty much automated. As I mentioned in the previous slide, you just have to load your samples and assays into that IFC, and you can do that using pipettes or using robots, whatever you have in your lab. After that process, everything else happens on the IFC. It's essentially walk away, and as such is load and go. Our microfluidic technology also spans a lot of different types of throughput. In other words, what you're able to achieve with microfluidics is not just ultra high throughput, and it's not mid throughput. You can even start from low throughput because we've got various different types of integrated fluidic circuits that can be used dependent on your throughput needs. And as I mentioned previously on the last slide, our technology does not come pre-spotted. And that's great for you. It's great for you because it means that you do not have to wait a long time to get our IFCs manufactured. It means you can take the individual targets that you're interested in studying and put them right on this chip or on this IFC. And you can add, replace, remove them as you see fit. Our content is not pre-spotted. So these three factors altogether, achieving greater cost effectiveness, scale sample throughput, and adding, replacing, or removing assays on demand, all help our IFCs to automate molecular biology in nanoliter reaction volumes. Let's take a look at some of the types of applications that these IFCs can be used for. These IFCs are generally used for all types of molecular biology applications. This includes genotyping, gene expression, copy number analysis, digital PCR, targeted and unbiased NGS library preparation. You'll see in the next few slides, we have a wide range of different types of IFC. In these coming slides, the nomenclature is important for you to understand. When we talk about these IFCs, the example here is the 4848 IFC. The first number here is the number of samples, and the second number is the number of assays. That nomenclature is similar across all of the IFCs that we have, and we'll come on to talk about them soon. In the meantime, let's have a look at how this really does save you time and save you reagents. And let's take an example of looking at our 9696 dynamic array. The previous slide showed that we had the 4848 dynamic array, but this example here is that of our 9696. And for those listening carefully, you'll understand that 96 refers to 96 samples, the first number, 96 assays, the second number. On this one device, we are able to assay 9,216 different reactions. That's a very high throughput approach. But as I mentioned, remember that we have approaches for all of the other types of throughput too. But this is a good example of someone who might be running quite a high throughput assay and needs to have that type of throughput and scalability. The other thing you'll notice on this slide is that the PCR reaction volume is nanoliter based rather than microliter based. So if we compare that to a 96 well plate, on the Fluidigm IFCs, we're running nanoliter reaction volumes versus microliters. What does it mean? It means that when you run a 96 well plate in your current lab setup, that reagent usage is 1,000 times higher on that 96 well plate than it would be running the Fluidigm. Think of the reagent cost that you can save if you're able to save 1,000 times using microfluidics. Also, the number of reactions on this IFC is much higher than what is being performed on a 96 well plate. On a 96 well plate, we're looking at 96 different reactions. With our Fluidigm IFC using the 9696 dynamic array, we're able to process 9,216 reactions. Plate throughput is 100 times lower than what is possible using our IFCs. To make this really clear, one of those IFCs is the equivalent of all of those 96 well plates that you've just seen appear on the right-hand side of the slide. On one IFC, we can take all of those 96 well plates. What that means is, and the key take home, is that one IFC is the equivalent of 96 well plates. Imagine running close to 196 well plates. Imagine the labor time. Imagine the cost. We save that to you with our microfluidics. If, on the other hand, the lab is routinely running 384 well plates, well, one of our 9696 IFCs is the equivalent of 24 384 well plates. So again, we're saving a lot in reagent usage, we're increasing the throughput dramatically, and we're saving you cost. Fluidigm microfluidics allows you to achieve significant savings. So let's take a look now at all of the different types of microfluidic devices that we offer. And Fluidigm is the market leader for microfluidics. And because of that, we've got a whole range of different types of IFCs that all work slightly differently to deliver different types of applications. The key take home on all of this that you're about to see is that no matter what the type of application, the Juno system and the Biomark system that we're going to talk about runs all of these IFCs. And you'll see that by investment into these platforms, all of these different IFCs and all of these different applications can be run. So let's take a look at the first type of IFC that Fluidigm worked up and built. It's a reaction partitioning IFC. What that means is that unlike most of our IFCs, the samples and assays are all mixed into individual inlets. Inlets are the little holes that you can see on the right-hand side of the IFC in view currently. So in this example, we have 12 different inlets. And so what that means is that the sample and the assay are combined together in one inlet. And in this particular reaction partitioning IFC, we use microfluidics to split that sample up across many individual PCR reaction chambers. A reaction partitioning IFC is generally used for digital PCR type applications. We'll come on to that soon. The other type of integrated fluidic circuit that we have is a combined reaction assembly and harvesting IFC. So rather than partitioning, where we split the sample up into different chambers, with our combined reaction assembly and harvest IFC, we assay in parallel. That's how you get the matrix that we mentioned at the previous part of the presentation. So in this example, again, you can see we have a 48-48 IFC. Because of that sample to assay metrics that exists, you're able to generate data for more than 2,000 reactions in that one IFC. And because of every sample seeing every single assay, it makes this IFC valuable for things like gene expression or high throughput fluorescence-based PCR. The harvesting capacity of this IFC, meaning that you can pull amplified PCR products off of the IFC, also makes it applicable for applications like library preparation for targeted next-generation sequencing applications. The last type of IFC is a capture, multi-step reaction chemistry, assembly, and harvest IFC. This is probably our latest generation of IFCs. And this generation of IFC is not a parallel loading with sample and assay. It's more independent valve control. And what that independent valve control means in real terms is that you can add reagents to different stages of the workflow. And that's very important. It's very important in applications such as single cell analysis, but it's also important in applications such as RNA sequencing library preparation. And we're going to really focus on RNA-seq library preparation towards the second half of today's webinar. So what does all that mean in real life? Yes, it means that we've got a whole range of different types of microfluidic devices. It means that they can be used for different types of genomics applications. But what specific applications can we help you with? On the left-hand side of this slide, you can see examples of our reaction partitioning. We mentioned that reaction partitioning is all about just splitting the chambers up. So on the left-hand side, you can see the types of applications that our reaction partitioning IFCs are used for would really be digital PCR-based applications. Unlike some technologies, the Fluidigm digital PCR applications monitor in real time. That means that in addition to you getting the endpoint yes or no answer, you also have access to the amplification plots. So it means that our digital IFCs with real-time PCR monitoring can be used for things like absolute quantification, copy number variation, haplotyping, rare mutation analysis, and CRISPR validation. The other application range is really key because this is the application range that enables you to go from very low throughput all the way to very high throughput, meaning that we're providing you with a very scalable base solution. Our combined reaction assembly IFCs are shown on the right-hand side of the slide. Because every sample in these IFCs is exposed to all of the assays, it makes them invaluable for real-time PCR-based applications. And those can be gene expression or quantitative real-time PCR, gene expression analysis, viral detection, very important in today's climate, copy number variation, SNP genotyping, as well as protein biomarker discovery. In addition to that, you'll notice on the previous slide, we also spoke about combined reaction assembly and harvesting. The and harvesting is missing from the PCR-based applications because in general, you do a reading on our Biomark HD platform, you get the signal, you report the result. The harvest part on our IFCs really relates to the library preparation part. Library preparation for next generation sequencing. And as you know, the reason that you have to harvest those products is so they can then go onto the Illumina or any other type of next generation sequencing platform. And so on the left-hand side, what you can see is that our combined reaction assembly and harvest really is for IFCs applicable for targeted DNA sequencing or targeted RNA sequencing applications for library prep. The right-hand side of the slide is going to show our multi-step reaction chemistries. Multi-step reaction chemistry being the latest generation of IFC, where we have independently controlled valves, which is important so that you can add reagents at the right time. This means that with our brand new 48.Atlas IFC, you can add the components to perform the RNA capture, you can add the components to perform the reverse transcription, the amplification in sequence. Very important because essentially what you're doing with that new generation of IFC is taking it from going to multi 96 well plates into one microfluidic device. And we're going to talk about this IFC later on in this presentation, where we talk about using the 48.Atlas IFC to capture and process the polyadenylated RNA from bulk RNA for RNA sequencing based library prep. Okay, so what we've gave you so far in this webinar is an introduction to the microfluidics. and hopefully an understanding of the different types of microfluidic device that Fluidigm can help you with. Let's talk a little bit about the systems, because we never really went into details on the systems earlier on in the presentation. So let's introduce them to you now. So Fluidigm systems are offered to be able to process the integrated fluidic circuits, or IFCs, that we mentioned earlier in this presentation. We have two preparatory systems. On the left-hand side, we have the Juno system. The Juno system will process and prepare any IFC that Fluidigm has on the market. Independent of scale, independent of throughput, independent of valve control, all of them can be ran on the Juno system. The C1 system on the right-hand side of the slide as a preparatory system is used to prepare single-cell based applications. So if we go back to the Juno system in terms of preparatory device, the Juno system processes all IFC formats. And that Juno system sits in front of our analytical platform. The analytical platform shown at the bottom left-hand side of the slide is our Biomark HD platform. This is the Fluidigm microfluidic real-time PCR-based platform, and it can be used for applications such as SNP genotyping or sample identification, gene expression, copy number variation, and digital PCR. The Juno system also sits in front of any Illumina next-generation sequencing system, and it sits in front of them because the IFCs that we mentioned a few moments ago can be used to prepare libraries for targeted DNA or targeted RNA library prep, or our most recent addition, unbiased library prep for RNA sequencing. The C1 system can also sit in front of the Biomark platform to perform automated single-cell capture and then targeted single-cell gene expression analysis. The C1 system also sits in front of any Illumina platform and is validated for applications such as RNA sequencing, immune profiling, DNA sequencing, and most importantly in today's single-cell market, multi-omic applications, such as analyzing RNA and DNA from the same cell or RNA and protein from the same cell. These systems altogether with the Juno, the C1 and the Biomark HD platform enable scalability, flexibility and workflow efficiency. They process collectively all of the Fluidigm IFCs. So in wrapping up this introduction, before we go into more detail on our new RNA sequencing application, the genomics-based applications by Fluidigm are unparalleled by any company. We offer solutions for targeted DNA library preparation, gene expression and viral detection, genotyping, molecular sample identification, copy number variation, digital PCR, single-cell analysis, and of course, RNA sequencing library prep. The rest of this webinar is going to be focused on RNA sequencing library preparation. So let's start to focus more on the Advanta RNA-Seq NGS library prep. Let's talk about this application. So what is it? So this application is all about providing a workflow to enable you to pipette RNA into our IFC and then prepare those individual RNA samples for next generation sequencing library prep. What that means is that on our IFC, you can pipette 48 samples into the IFC, and then the IFC, using the multi-step reaction chemistry that we mentioned a few moments ago, will then perform the RNA capture, the polyadenylation capture. It will then perform the reverse transcription. It will then perform the amplification and barcoding. And then at the end of the workflow, you're able to take the amplified products off of this, quantify them, and put them into the Illumina sequencing platform. This workflow is a very well automated workflow. It takes all the labor out of library prep, and it also reduces the cost by about half of what you would be doing if you were doing this in a 96-well plate using liquid handling robots. So we'll go into that in more detail in the next few slides. But for now, takeaway message is simple. The Juno system on the left-hand side is the capital equipment piece that's required to run our integrated fluidic circuit. So the Juno system is a next generation sequencing library prep instrument, as well as a universal controller that we mentioned on the previous slide. And it will help with cost efficiency and enables you to walk away. And it enables you to program when you want your PCR amplicons to come off of the chip. That's very important because it means that you're no longer tied to the bench in a 96 well or a 384 well plate. The device in the middle of the slide is the 48.Atlas IFC. Remember again that this IFC is not a metric-based IFC. It's an IFC that runs using our multi-step reaction chemistry. And what that means to you as a user is that you pipette RNA into that IFC and you get library out of that IFC. And hands-on time is only 30 minutes. Compare that to your existing workflows for RNA-seq. And the last part on the right-hand side of the slide is our Advanta RNA-Seq NGS library prep reagent kits. For the Advanta RNA-Seq NGS library prep workflow, Fluidigm provides you with an end-to-end solution. Once you've got your libraries off of the Fluidigm platform, they're ready to be quantified and they're ready to go onto the Illumina sequencing platforms. So, key takeaways from this application at a high level, before we start to go into some more of the details, is that we're providing you a system here that does a walk-away automation. You pipette your samples into the IFC, the Juno system does the rest. That means that we are creating a highly efficient workflow, maximizing lab resources, minimizing the hands-on time, and reducing the cost of reagent consumption. Because again, on this IFC, we are performing at nanoliter reaction volumes versus the microliter reaction volumes that you would perform on 96 well plates. And as we mentioned earlier in the presentation, we use this multi-step reaction chemistry, meaning that each chamber is independently controlled, which means that only the reagents required for a particular step are applied to the sample. And because of the way in which the microfluidics work, we have very high efficiencies because the low volumes, we're actively mixing on that IFC all of the time. So on this IFC, we have a very robust chemistry. Reverse transcription is using random primers and template switching to generate full-length stranded libraries for up to 48 samples. So let's take a little bit of a look of the Juno workflow and the Advanta RNA-Seq NGS library prep workflow. So as I mentioned, there's multi-steps to this, but once you have pipetted your samples and reagents into the IFC, everything else is automated. So what you can see on this slide at the left-hand side is that you load your RNA, reagents, and oligo-DT onto the IFC. And that sample loading will take around 60 minutes. Once that's complete, it then goes into the Juno platform for automated IFC prep and processing. And that takes place for around 9 hours. But don't be scared about that 9 hours. You don't have to be there all that time. That can also be run overnight, and you can program the Juno system to tell it when you want the amplified libraries to come off. So if you just finish your shift at 7 p.m. and you don't want to be there till the early hours of the following morning, you can tell the system, hey, I want to harvest at 10 a.m. and it will have it ready at 10 a.m. for you. Once that harvest is complete, you then pull the samples, takes around 10 minutes, clean up, perform the adapter, PCR, and QC, and then they're ready to run on any of the Illumina next generation sequencing library prep systems. Here's an example of the IFC to show you the pipetting maps that are required. As I mentioned, these IFCs can interact with any of the pipettes that you have in your lab, but they even interact with liquid handling robots. And we have customers that use them for that. And that really helps it be a fully automated workflow. Just to mention a little bit more about the multi-step reaction chemistry. This is very important for you to understand. And it's important for you to understand so you can really see the workflow efficiency that is happening on this IFC. So what you can see here on this slide is that the sample is going through this line, and these little gray bars that you can see here, 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 and here are all independently controlled valves. That's important because it means that whatever is happening in this chamber marked 5 nanoliters, which happens to be the bead column formation, polyadenylated RNA capture, washing and eluting. Whatever's happening in that chamber is contained in that chamber. That's important because it means that with that containment, the efficiency of the reaction is much higher. Once that's complete, the valve will then open up and will perform the fragmentation, followed by reverse transcription, followed by the barcoding PCR, followed by the harvest. Look at the volumes on this slide: 5 nanoliter, 55 nanoliter, 70 nanoliter, 360 nanoliters, all very small volumes, meaning that you're saving a lot when it comes to reagents. In general, comparing our solution to a 96-well plate or a 384-well plate using larger volume chemistry, we're about half the cost. So we're saving you time, we're saving you effort, we're saving you labor cost. That's what the 48.Atlas IFC is all about. And for those that need a little bit of clarity on that, here's an example of that. What you can see on the left-hand side is how the Advanta RNA-Seq library prep kit workflow compares to the Illumina TruSeq standard mRNA-Seq library prep kit workflow. The key take-home on this slide is look at all of the different steps that exist with the Illumina platform. And look at how the Fluidigm with the Juno and the Advanta RNA-Seq library prep kit automates that. So let's look at that in detail. The teal color is the hands-on time, 1 hour hands-on time. When you then run that on the Juno and with the Atlas 48 Atlas IFC, 0 hour hands-on time. 9 hour runtime, but no hands-on time. At this stage in the workflow, the lab personnel can be away doing something else. That is then followed by some bench work, which takes around about an hour. Again, look at the comparison. Compare the teal on the left-hand side to the teal on the right-hand side. 4 hours hands-on time up here for Illumina, 2 hours 45 minutes hands-on time post-workflow. And then the bar graph in the center of the slide is showing the hands-on time for the Advanta RNA-Seq library prep kit versus the TruSeq kit. You can see that the hands-on time for the Illumina solution in plates manually is 6 hours 45 minutes. The Fluidigm automated microfluidic approach is 2 hours. Think of the cost saving, the labor saving, and the hands-on time saving that the Advanta RNA-Seq library prep kit can provide you. What about throughput? What does this mean to your laboratory in terms of throughput? So each of those 48.Atlas IFCs runs 48 samples, as you can understand from the name. An example of a lab working 5 days a week processing these IFCs can be set up a little bit like this. In other words, if you take day 1, you have the morning off, but day 1 in the afternoon, you can prepare the IFC 48 samples in an hour, run the Juno overnight, get the data off in the morning of day 2, following the second IFC in the afternoon, followed by the third IFC on day 3, followed by the fourth IFC on day 4. What that means is that in 5 working days, you can process 192 samples. Why is that important? It's important because if you run a core lab and the samples come in day, maybe one day you have 2 samples, the next day you have 3 samples, you can batch them and run them on these IFCs. So in one 5 working day week, you can process 192 samples using our 48.Atlas IFC. The other thing I mentioned earlier in the presentation was the savings that this kit allows you to have. And this slide clearly shows you the savings that you get when you compare the microfluidics platform, which is described in this graph as Advanta, compared to the TruSeq protocol, which is great. So if you look at 2,500 samples, the cost of the Advanta RNA-Seq kit is around 82,000. If you compare that to Illumina, it's 175,000. But what you'll see is that more samples that you run, the more that you save. So in other words, if you have a 5,000 sample project, by using Fluidigm Microfluidics and the Juno platform, you're saving $185,000 in reagent. If you expand that from 5,000 to 10,000, you're actually saving around $370,000 when you use the Advanta kit compared to the TruSeq kit. And as I mentioned, the workflow is pretty straightforward. Most of it takes place on the IFC. So on the left-hand side, you can see all of the steps that take place on the IFC using the Advanta chemistry. IFC performs the capture and the polyadenylation of the RNA using the beads. It performs the elution, it performs the reverse transcription and template switch, and it performs the sample barcode PCR. On the right-hand side, you can see what takes place off IFC: harvesting, pulling, amplifying, and then right on to the Illumina sequencer. Our product specifications are shown on this slide, and more information can be found on the Fluidigm website under the Advanta RNA-Seq NGS library prep kit part of the website. But again, just some key levels here. We have this protocol for full-length stranded libraries from random primed polyadenylated RNA. It runs on total RNA isolated from tissues and cell lines. It's best when you have RIN scores of greater than 7, but you'll see later in the presentation we've also looked at lower than 7 and it works just fine. The input amount is between 10 and 100 nanograms. It runs on the Juno instrument with what we call the TX interface plate. That's the plate that sits on top of the IFC. And it runs on our 48.atlas IFC. The rest of the information on this slide is pretty self-explanatory based on the previous slides presented in this webinar. So let's take a look at some of the data. How does this actually compare to the TruSeq application in Fluidigm's hands? And then also let's take a look at how it's working in our customers' hands. As I mentioned earlier on in the presentation, we're going to show you some data from Scott Magness's lab out of the University of North Carolina. We won't go through all of the details on this slide, but it's there for your reference on how we validated this analytically from an internal perspective. The key take-homes are shown in the subsequent slides. So what you can see here is a summary of our performance results generated from the analytical validation testing. You can see that when we compare universal human RNA reference to brain, you can see the reads mapped to the genome. You can see the percent mapped to the transcriptome, known as the RefSeq. You can see the percentage ribosomal reads. You can see the percentage unmapped reads. And then you can see the correlations. The key take home on this is that we are getting a very high percentage of reads mapped to the genome. We're getting a very high percentage of reads mapped to the transcriptome, and we're getting a very low percentage of ribosomal reads, as you would expect. What this means to you as a user is that the smaller volumes, the automated workflow, the low hands-on time do not compromise the integrity of the data. Also, when we check read uniformity from the pools that we get from these individual libraries, we are getting very good read uniformity. You can see on this slide that all libraries have achieved the target read uniformity that we expect in this workflow. And further, when we think about the technical reproducibility between different operators, many of you work in core labs, many of you work in research labs, where there's many different types of work going on. You have to be able to check that actually our gene level Pearson's correlation between operators and between different concentration is sound. And it is. On average, we see a greater than 99% concordance observed, showing that our system and our workflow, i.e. the IFC, the Juno, and the reagents are operating with excellent robustness. And you can see that both in the universal human reference RNA, you can see that in the brain, and you can see that across different concentrations, 10 nanograms versus 100 nanograms, operator 1 versus operator 2. We also did some comparisons of our Advanta RNA-Seq kit to the Illumina TruSeq kit. And again, for reference, this was the experiment design that we performed. Let's take a look at some of the data and the take-homes. So we're able to see when we do a dynamic range per sample and input amounts using the ERCC spike-in controls, when we take ERCC mix 1 and mix 2, and we compare that to the average read count and compare that to the Advanta average read count, at 110 nanograms, we are getting comparable spike-in detection. So that comparable spike in detection is happening whether you use the cumbersome TruSeq based approach or whether you use the highly efficient microfluidic based approach from Fluidigm. And I'm sure you'll agree it's better to use the highly efficient approach and get the same data than using the less efficient approach. And let's compare how these genes detected per sample also looks. Again, this is us comparing the Advanta RNA-Seq to the TruSeq reagents. And again, Advanta shown in purple, TruSeq shown in gray. You're seeing that really you're getting the same numbers of genes detected compared to the Advanta and the TruSeq in the universal human reference amount at both 10 nanograms and 100 nanograms. And it's the same story in brain. So again, we're achieving comparable gene detection across different samples, whether you're using the Advanta RNA-Seq application or whether you're using the TruSeq application. And of course, using the Advanta RNA-Seq application helps you get there faster, more efficiently, and saves you a lot of money. And if we look at the genes detected per transcript, it's also comparable. You see where the story's going. The gene detected per transcript is also very similar in the universal human reference RNA and the brain at both 10 and 100 nanograms. And between kits, of course, we see the same good, strong concordance, R value of greater than 0.94. That's really, again, showing the value of the robustness of the workflow, the chemistry, the chip, and the system. I mentioned earlier on when we were looking at the data that we also looked at different RIN values or RNA integrity numbers. And, we say in our performance criteria, above 7 is best. Of course, we live in the real world and we know that core labs have different types of sample and different types of RIN value. So you can see that Fluidigm have created this beautiful bank of different tissues across different types of RIN value. So again, you understand the impacts of RIN or lower quality and what impact that will have on the number of genes and the number of transcripts detected. So again, living in the real world, this system is usable with varying RIN scores. It's usable with varying tissue types. In the last couple of minutes of the presentation, I want to also show you some real-world example. Real-world example that we got with our collaboration from a customer at the University of North Carolina. This work is all about the analysis of transcriptome in hypoxic intestinal stem cells. The data comes from our collaboration with Scott Magness. And Scott Magness has a lab at the UNC that really focuses on long-standing interest in intestinal stem cells and the genetic mechanisms that regulate ISC differentiation and maintenance. And Scott's lab and Scott's research really relies on working with cell inputs that are quite low. And they're in different types of oxygen concentration. So the samples are not always the most robust. So Scott was really looking for a highly efficient workflow. He was looking for something with low hands-on time that does not compromise his data. And that's why he selected the Fluidigm 48.Atlas IFC and the Advanta RNA-Seq NGS library prep kit. Let's take a look at some of Scott's data. So Scott was all about analyzing the transcriptome in hypoxic conditions. So the Magness lab did actually develop a microfluidic device that actually enables the cells to sit in a chamber and enables them to be exposed to different concentrations of oxygen. So the experimental parameters were that Scott was going to analyze or has analyzed 24 samples exposed to different hypoxic conditions and overcome the challenges mentioned on the previous slide with the input of RNA and the need to analyze many replicates. Scott adopted the Advanta RNA-Seq NGS library prep workflow, where he took the samples from his gas chip, pipetted them into the 48.Atlas IFC, ran them on the Juno, then processed on the Illumina platform. The sequencing metrics across 24 samples showed that 98.5% aligned to the genome and 80.2% aligned to transcriptome. Very good parameters and in guidance with what we've saw from our internal data. Again, when we look across these performance metrics, you can see the total reads per sample, you can see the percentage of reads unmapped, the percentage of reads aligned to the genome, the percentage of reads aligned to the transcriptome. Again, the key take home is that these are high mapping statistics with a very low percentage of reads derived from ribosomal RNA sequences. It's showing the value of our microfluidics, and it's showing that you get very good sequencing metric data from these microfluidic devices. So Scott was able to generate principal component analysis, clustering his samples at the different times in which they were exposed to different levels of hypoxia or different levels of oxygen. And you can see that across 6, 24, and 48. Scott was able to also create these volcano plots to detect time-dependent changes in gene expression from 6 hours through to 48 hours. And further, Scott is able to then perform Venn diagram analysis so that he can see the characterization of gene expression changes across the different exposure times in each of his sample sets. In this study, Scott, which is now ready to be published, or the manuscript is being created, was able to study the duration of hypoxia on ISCS induced differential gene expression at each time point measured by RNA-seq. And Scott and the team at UNC were able to do that using the Advanta RNA-Seg NGS library prep kit. Because they had low input of RNA, they got highly reproducible results, and the workflow is highly economical. I really hope that you found today's webinar useful. I hope that you've got something out of it. And at this point, we'll take any questions that may have came in through the chat box.